the topic was preparation and staining of a blood smear, and after staining a blood smear, the cells had to be identified, then the cells had to be counted. The topic which you are to face in the exam is differential leukocyte count. You have to count cells and write the results, and then you will be asked questions on that. This is how the hematology section, the exam, how it is conducted. Hematology section, the exam usually out of 100 marks, that is up to the institutions, 10 marks are allotted. Out of 100 marks, I told 40 for VIVA and 60 for the practical part. Out of that, hematology 10 marks, okay, clinical 20 marks. This may change, but usually it is like this. Hematology 10 marks. So for that 10 marks, you have to do one or more experiments. If it is a long experiment which takes 40-45 minutes, 1 hour, then usually you are to do one experiment and 10 marks are allotted for that. If it is a short experiment, then maybe 2 or more, okay. So for hematology, we have these, except differential leukocyte count, we also have total leukocyte count, total erythrocyte count, determination of blood groups, okay, estimation of bleeding time and clotting time. These topics are there. So these topics you have to learn in the hematology sessions. So now the topic which is going on is differential leukocyte count. For that, we need glass slides over which a blood smear will be prepared. And for preparing the blood smear, we need blood, obviously we need blood, that is usually capillary blood or venous blood. For few experiments will be there, differential leukocyte count is possible with venous blood. Few experiments will be there, 1 or 2, that which is not possible without capillary blood. So from the first day itself, it is recommended that you collect capillary blood. And for collecting capillary blood, from where do we collect the blood? We collect the blood usually from the fingers. It can be collected from the earlobes also. In case of children, it can be collected from the heels. So from the fingers, from your own finger, you prick with a needle which is supplied here, prick with a needle, and there are precautions to be followed while doing this. So obviously, which comes to our mind first, one needle has to be used by one person only, exclusively. Needles cannot be shared, okay, and it should be used, recommended that not more than one time, although it may be done, so not multiple times, but 1 or 2 times. So pricking the finger, for pricking the finger, you have to, those who don't know still, you have to use cotton and some alcohol based liquid, spirit, and rub the area first. Usually we prick just this part of the finger, this part, ring. Usually we use the ring finger or the middle finger and prick with a needle so that capillary blood comes out. Capillary blood comes out and it is freely flowing. So there is difference between freely flowing and oozing out, okay. Blood should be freely flowing. Otherwise, what is our tendency when we have to collect blood? If blood is not freely flowing, we usually try to squeeze the finger and then take the blood. What happens is that while squeezing the finger, instead of blood, mostly what comes out is tissue fluid. So tissue fluid will not help. So after pricking, the term is used a single deep prick, so that capillary blood is freely flowing out, which can be taken on a glass slide, okay. Now how to take on a glass slide, those who still don't know, we'll do it in the lab. We have to keep the slide on a horizontal surface, and how to prepare the smear with the help of another slide after taking the blood drop, we'll just do it in the lab. Those who have still not seen, will help you, and those who have already seen, who have been newly admitted, take their help also, and you have to learn. And I will recommend always during these practical sessions, at least the hematology sessions, you should have the books. I always recommend that. Don't come to the class without the books. At least you will make a batch of 3 or 4. After this, you will get your roll numbers and attend the sessions. According to the roll numbers, we will divide in these groups, say 3 or 4. So at least 1 or 2 books you should have, so that you can refer to the books while doing the experiment in the lab. You can refer to the books if it is needed. Obviously, referring to the books is not allowed during the exam, but during learning you have to refer to the books. So any standard textbook of practical physiology you have to follow. I am coming to that. Now, taking the blood on the slide and preparing the smear with the help of the slide, those who still don't know, I am not, it is mostly no use just verbally speaking it out. You have to do it and learn. So, we will do it in the lab, how to take that smear. After preparing the smear, allowing it to dry, it has to be stained. So, it has also procedure, how the stain is to be poured drop by drop, covering the smear, and then waiting for 2 minutes, and after that putting distilled water or buffered water, whatever is available, and that has a particular quantity. That means Leishman stain, you have to count the drops, how many. Leishman stain over use over here. Leishman is the name of the stain. Okay, nothing to write. I am going a bit fast. Already these had been discussed, but I am going just revising for the students who have already attended, and for who have not attended this topic, it is new, but we will learn it over there directly. I am just telling a few words on it, that it has to be Leishman stain has to be taken. The name of the stain is the Leishman stain. Okay, there are other stains, James stain, Wright stains, not available over here. So Leishman stain we use, that we have to put drop by drop over the blood smear so that it covers the smear, and then after that, after waiting for 2 minutes, and each step has a reason, waiting for 2 minutes has a reason. Okay, that is the fixation time. After that we put distilled water or buffered water. If it is distilled water, the number of drops of distilled water to be used is double the drops of Leishman stain used. If it is buffered water, same number of drops as the Leishman stain. Then for actual staining, we keep it for 10 minutes. Then whatever there is extra fluid over there, we wash it away very gently. That has also how to wash it away. If we wash it away not gently, if the tap, if very fast, if we use the tap water which is falling from the tap very fast, then the smear may be washed away. So very gently to wash it using our own fingers, allow the very gently tap water is allowed to fall on our finger, and then it washes away the scum over the smear, extra fluid portions come over the smear, and then after drying it is examined under the microscope. So for examining under the microscope, I think all of you have an idea of the microscope. We had a class on the microscope also, but microscope, it is expected about the microscope you know from your school days. You have already used a microscope, but how to handle a microscope, be very careful. We will show it in the lab, how to handle a microscope, and the parts of the microscope we have to know how to use them, what lens is to be used for which experiment, what magnification of the lens for which experiment. Like for this experiment, differential leukocyte count, we usually use the objective lenses which are either focusing it, magnifying the object 45 times, 40 to 45 times or 100 times. These two are used. There is a lower one than this, that is magnifying it by 10 times. That one is not used for this experiment. That one is used for other experiments. So for this, either the 45 which is magnifying it 45 times or the 100 times, and the total magnification will be, there is one lens called the eyepiece lens, we will see in the microscope. So the total magnification will be, if two lenses are there focusing on the object, total magnification is multiplying one by another. So eyepiece lens is usually magnifying the object 10 times, and if the objective lens is multiplying it 40 times, then it is 10 into 40, it is 400 times. If the eyepiece lens is 10 times and that is objective lens is magnifying 100 times, the total magnification is 10 into 100 is 1000 times. The object is being magnified 1000 times. And for using the objective lens, objective lens of 100, which has 100 magnification, for this experiment to identify the cells, we use oil, okay, cedar root oil. Cedar root oil, which is usually used depending, that is that particular oil is used because of the refractive index, okay. So this is how it is done. We the details of how it is done we will do directly in the lab. So after this, when examining the smear, we have to find cells, okay, identify cells. So what are the cellular components of the blood? These are very basic question. RBCs, WBCs and platelets, red blood cells, white blood cells and platelets. Here in differential leukocytes are WBCs. Differential leukocyte count means the percentage of the different types of WBCs among the total number of WBCs, and it is expressed as a percentage. So, if we can count 100 WBCs, how many are neutrophils? The types of WBCs are neutrophils, basophil, eosinophil, monocyte and lymphocyte. So, three of these types are granular cells, and two of these are agranular cells. They are called agranulocytes, granulocytes and agranulocytes. So, how to identify them? We will identify under the microscope. Just a few points. Most of the cells when we are examining under the microscope, most of the cells will be RBCs, obviously. As we know, they are huge in number compared to WBCs and platelets. So, what is the theoretical count? We know 4.5 to 5.5 millions per microliter of blood, microliter or millimeter cube of blood, microliter or millimeter cube. 1 millimeter cube and 1 microliter speak of the same volume. Okay. So, one is in the SI system, one is in the CGS system. The microliter is 10 to the power minus 6 liter. Millimeter cube means cubic millimeter. Some books it is written microliter, some books it is written millimeter cube, but it is the same thing. So, per millimeter cube of blood, it is 4.5 to 5.5 million, whereas WBCs are only 4,000 to 11,000. This is the standard number accepted theoretically. So, you see it is almost 1,000 times RBCs. So, most of the cells which are viewed and after staining, without staining we cannot see any cells. So, after staining, these are the RBCs. So, using the Leishman stain, these RBCs appear as uniform non-nucleated discs. Obviously, RBCs do not have a nucleus. So, the cytoplasm, there will be a cytoplasm and a cell membrane. So, inside the cell membrane there will be cytoplasm. Using the Leishman stain, we see the RBCs as non-nucleated discs which are pinkish in color, because RBCs are stained by the component of Leishman stain which is called Eosin. Okay. So, Eosin stains the RBCs, and it also stains the granules of Eosinophils which are coarse granules. Okay. So, difference between fine and coarse, that we can find out just by, this is by experience, just by looking at those cells. So, when I am speaking of these things now, what are the structural features, how to look at, I am just visualizing. As you go on visualizing, this will automatically come to your mind while reading the description. For the first 1 or 2 times, you have to just orient yourself that this cell looks like this cell looks like this, and then you can visualize even when not looking at the cells actually. So, RBCs appear as pinkish discs, non-nucleated, stained by eosin. Cytoplasm is stained by eosin. Eosinophils, the granules will be stained by eosin, and they are coarse granules, and eosinophils are a type of WBCs which have a nucleus. The nucleus is bilobed nucleus, and the typical description for it is spectacle-shaped nucleus. Spectacle-shaped nucleus, bilobed. Nucleus of neutrophils, basophils, eosinophils have lobes. Of these granulocytes have lobes. So, eosinophil is bilobed nucleus, and the typical shape is spectacle shaped. Okay. In medical science, everything is not always perfect. Okay. In medical science, everything cannot be perfect, but it is almost more or less typical spectacle shape. Some may be just somewhat distorted, but eosinophils typically spectacle shaped bilobed nucleus with coarse granules which are stained by eosin, that means pinkish granules, but the nuclei will be, what is the other component of the Leishman stain? Components, different components are there, but which staining components are eosin and methylene blue. The nuclei will be stained by methylene blue of the neutrophils, of the eosinophils, of the basophils. Now, what I would suggest, when you examine, look for the cells and comment which cell is this, you don't go for basophil, at least as beginners. In this first phase, you don't go for basophils because basophils are very rare. Those who already know, basophils are very rare. If you count 100 cells, maybe there are maybe no basophil or one. If you go on counting say 1000, 5000 cells, then only you may get rarely some basophils. So theoretically accepted value of basophil is 0 to 1%, 0 to 1%. Okay. Then there are monocytes, eosinophils are in the range. Eosinophils say 4 to 6%, monocytes say 2 to 4%, like this. They are also not very huge in number, but at least few can be found. Maybe 2 or 3 can be found on counting 100 cells. In some cases 6 or 7 can be found, but basophils are very, very rare. Although we should know about the structural features of the basophil, I would suggest that don't comment. If you comment any cell as basophil, you have to stand it, that why you are calling it a basophil. So it is better not to suggest. Okay, leave that field in every high power field. High power means when you look at it under the magnification with 40 objective lens with magnification 40. In every high power field, ideally it is told there should be 1 WBC. Okay, if you by any chance if you find that it is a basophil, I would suggest, and I am saying it on record, just ignore it and pass on to the next field, because it is difficult to stand, because basophils are very rare. So maybe the examiner will tell you, the person who is conducting your exam, your examiner will stand some other cell and then you will be wrong. So neutrophils, eosinophils, lymphocytes and monocytes will be rather easier to locate and comment on. So how are neutrophils or eosinophils? I was telling about eosinophil, that what are the structural features. Now before that, you compare the cells, before just looking at the structural features. Whenever you find WBCs, WBCs are distinct from the RBCs. RBCs are just pinkish discs. There is no tinge of blue, purple, anything over there. So because it is cytoplasm, it contains no nucleus. Cytoplasm is stained by eosin. So these are pinkish discs, pinkish rounded discs. But WBCs, as the nucleus will be stained by methylene blue, it will take a purple, deep purple color. Okay, so WBCs are easily identified among the RBCs. And if those who have still not seen, if any of you are there who have still not seen under the microscope, then just even before seeing, try to visualize it. So many pinkish cells are lying, pinkish rounded discs are lying. Within that, maybe in one field, 1 or 2 or maximum 3 WBCs. Okay, so these are easily identifiable that this is a WBC. And also some platelets can be seen. Forget about platelets for now, concentrate on RBCs and WBCs. So what will be you first compare the size. All the WBCs, most of the WBCs at least, will be larger than the RBCs. Although RBCs are huge in number, most of the WBCs, the cell size of the cells larger are the WBCs. So neutrophils, eosinophils, basophils, although I am suggesting not to ignore the basophils. I have not, just comment, I have not got any basophil, but neutrophils, eosinophils, basophils, whatever you comment on, they will be 1.5 to 2 times, maybe around 1.5 times, and these are not exact. One cell may be smaller or larger, but it is roughly 1.5 to 2 times. And one thing, remember, don't go by theoretical values. Okay, just don't comment, at least for the exam purpose, because under the microscope we have to compare the sizes of the cells with one another. What we know theoretically, RBCs are diameter of RBCs, and we will know, it will be discussed in the hematology theory sessions. Diameters of RBCs is 7.5 micrometer, probably you know 7.5 micrometer. So this particular WBC is twice, so it is about 14-15 micrometer. But you cannot measure it under the microscope. There you have to compare the size of the WBCs with the size of the RBCs, and WBCs are more or less, say neutrophils, eosinophils, basophils are 1.5 to 2 times roughly the size of the RBCs. Okay, so in the pool of the RBCs, so many RBCs, you will find that cell is somewhat larger. And which cells are the largest? They are the monocytes. Okay, between monocytes and these granulocytes, which are the neutrophils, eosinophils, monocytes is one of the agranulocytes. Monocytes don't contain granules, lymphocytes don't contain granules. Large lymphocytes are almost more or less the size of the monocytes or somewhat lesser. Large lymphocytes. Only among all these cells, we classify lymphocytes according to size again. Some lymphocytes are small, some lymphocytes are large. Small lymphocytes may be somewhat larger than the RBCs, almost the size of the RBCs. So those are lymphocytes similar looking to the larger lymphocytes, but they are almost the size of the RBCs. All other cells, all other WBCs will be greater than the size of the RBCs usually. So neutrophils, and how to now after this, after comparing the size, okay, this WBC is, so this is a WBC, size is larger than the RBCs. What are the other points to note? Other points to note are the structural features of the nucleus and the granules in case of the granulocytes. So eosinophils will have coarse granules, neutrophils will have fine granules. Now this is when we are studying it theoretically, we are speaking of theoretically, we can, or we are writing the differences between the, we are thinking of the differences between the neutrophils and eosinophils, we just write coarse granules or fine granules. But what is the actual, how to look at it, it will come by how to identify, it comes by experience. Now eosinophils, how we will identify the coarse granules? So whenever you look for the spectacle-shaped nucleus and the granules, you will find some coarse granules, so somewhat not so fine, so they are coarse and they are stained by eosin, and you can identify. Within 1 or 2 days, you will identify that these types of cells are eosinophils. They will have a cell membrane, inside there will be cytoplasm, there will be cytoplasmic granules which are stained by eosin, so pinkish, so coarse granules, and the nucleus is purple and it is bilobed spectacle shaped. Comparing with neutrophils, which are the most abundant among the WBCs, theoretically it is 60 to 70% of WBCs are neutrophils. So the most abundant in among these fields, obviously much less than the number of RBCs, but among the WBCs highest number expected is neutrophils. Okay, so neutrophils are, except in pathological diseased conditions. Okay, except means sometimes in diseased conditions other cells will increase, other types of WBCs will increase. Like one example is in allergic manifestations, eosinophils increase. So still it will not increase more than the number of neutrophils, that, but if we count 100 cells, maybe 20 eosinophils are found. But normally in physiological conditions, not in disease conditions, pick it up, not in disease conditions, it is maybe 4 to 6, maximum 7 like this, eosinophils. Maybe it is 20 in some allergic manifestations. So if we draw the blood and examine under the microscope, maybe more eosinophils are found, but still it is not expected that it will exceed the number of neutrophils. So neutrophils are the most abundant of the WBCs, and granules are fine, and nuclei are similarly lobed as in case of eosinophils, but nuclei may be 2 lobe to 7 lobe, 4, 5, 6, 7. Till now we know of maximum 7 lobes of the nucleus. So 2, 3, 4, 5, 6, 7. It may be either 2 or 3 or 4, 4. So if you see multiple lobes, first think it of it is a WBC, greater in size 1.5 to 2 times roughly the size of the RBCs. The granules are fine, not comparing with the eosinophil, not as coarse, and the nuclei are say 5 lobes. So and neutrophils are most abundant, so confidently identify it as a neutrophil. Okay, so neutrophils and eosinophils are identified like this. In case of lymphocytes and monocytes, what will be lacking from these granules will be lacking, other than these neutrophils and eosinophils. Basophil, it is better not to comment. So neutrophils and eosinophils, comparing with neutrophils and eosinophils, granules will be lacking, and otherwise also these lymphocytes and monocytes are more or less easy to identify. Why? Because in case of the lymphocytes, cell membrane will be rounded margin, and inside the nucleus, a rounded nucleus, it is not exactly round always. Again, I am repeating, in medical science everything is not perfect, that whatever you expect will perfectly like that, but more or less it is rounded and it almost covers the whole of the cell. Okay, any problem? Okay, it almost covers the whole, don't talk in the class, almost covers the whole of the cell unless you are not told to. Okay, demonstration sessions are interactive sessions, lecture sessions are usually not interactive sessions. The faculty will speak and the student is expected to listen. Different teaching methods are coming up these days. These are being discussed for the last few years. Okay, so demonstrating sessions are interactive sessions, but in the class you are not allowed to talk among yourselves. That disturbs the class. If you have something to tell, just raise your hand and you may speak out. So lymphocytes and monocytes are easy to identify. Rather, the nucleus of the lymphocytes will be covering almost the whole of the cell. So it looks more or less like this. If it is a cell membrane, then almost the whole of the cell will be covered by a more or less rounded nucleus. So what lies this some cytoplasm just outside the nucleus, and this is just this term is used classically, thin rim of cytoplasm outside the nucleus, and this will be again deep purple like that, the nucleus. So that part, so don't go by this. This is just I am drawing by the hand. You will look at the cells and you will find pictures in the books, good pictures in the books, textbooks. So just I am trying to explain to you that it is easy to identify. It will be a rounded cell with a cell membrane. Nucleus will cover almost whole of the cell. In some cases it might be a bit smaller, but still it will be a lymphocyte, because other features are there. There will be no granules. Whatever cytoplasm we can find, there will be no granules over there. Okay, so the nucleus is a rounded one, almost covering the whole of the cell. And now what did we say few minutes ago? That is, lymphocytes may be either only of the WBCs, which are classified again according to size as small or large. So if this type of cell you find much larger than the RBCs, then it is a large lymphocyte. And if it is more or less the size of the RBCs, somewhat lesser in size than the neutrophils, it is a small lymphocyte. Again, during at least for for the first phase of MBBS students, it is not required to classify these are large, these are small. You just simply classify them as lymphocytes and write your count. Okay, lymphocytes, and what is left is the monocytes. Monocytes are also easy to identify. Again, the rounded cell. The nucleus in the case of monocytes, it is typically called, it is classically called bean-shaped or kidney-shaped nucleus, and it will lie towards one side of the cell. One side meaning like this. If this is, so say this is a bean-shaped, say this one side of the cell. So what happens is that this part is spoken of as extensive cytoplasm. That means a lot of, in case of the lymphocyte it is called as thin rim of cytoplasm, but as the nucleus in the monocyte is towards any one side of the cell, rest of the cell is the cytoplasm of the cell. And what feature is there in the cytoplasm, or actually lack of any feature, that is lack of granules. There are no granules. So, and this is, so lymphocytes or monocytes are rather easily identifiable. Confusion is there among neutrophils, eosinophils and basophils. Better not to comment on basophils, as there are very, very few in number, very, very few. So better not comment. So differentiating with the neutrophils and eosinophils, first by the size, almost they will be of the same size, 1.5 to 2 times of the size of the surrounding. Most which are lying in the field are the RBCs, but go for granules and nuclei, lobes of the nuclei. Now one question that may come to your mind, that is, eosinophil is bilobed and neutrophil is also maybe bilobed, maybe 7 lobed. If it is 5-6 lobes, it is a neutrophil, because 5-6 lobes are there not in the eosinophil. But if it is bilobed, so there may be a confusion. So try to identify the spectacle shape, which will come by experience. Once you start looking at some eosinophils, you will get an idea what a spectacle shapes look like. So spectacle shape bilobe nucleus is eosinophil, and simple bilobe nucleus, but not that typical spectacle shape, it is neutrophil. And also there will be the difference in the granules. Now one thing is, in the books it will be written like this, that which part of the cells, it is written the information that which part of the nuclei, which are the components of the different cells stained by which component of Leishman stain. So methylene blue stains these nuclei, eosin stains these. So just to remember it easily, just remember that eosin stains cytoplasm of RBCs and granules of eosinophils, and rest anything that you can think of in this context is stained by methylene blue. So don't try to memorize like this. It is so many things you will have to memorize in the coming 1 month, 2 months, 9 months, 4 years, and actually the whole life. Medical science, a lot of things you have to memorize, lot of things, because names you have to memorize. So what I suggest is whatever can be memorized rather easily. So to memorize easily, sometimes we use mnemonics. Okay, we know what mnemonics are. As we go on with the syllabus, there will be various examples. Give one example of what mnemonics are. Those who said yes. Yes, whenever we speak of something, we use mnemonics, means we use some, that means some trick or some common terms we use which we can relate with the actual topic. Those are called mnemonics. Like, you know how many pairs of cranial nerves are there in the human body? 12 pairs of cranial nerves, 31 pairs of cranial nerves, spinal nerves, sorry. So 12 pairs of cranial nerves have 12 distinct names. So there is one sentence. That sentence is not related to any way to medical science or biology. So there is one English sentence, the first letters of which that is the first letters of the cranial nerves and the first letters of the sentence matches. So what we do is remember that mnemonic and try to take that first letter and try to remember the name of those nerves. So this is what an example of a mnemonic is. Instead of memorizing so many things which are unfamiliar, we try to memorize some familiar thing and try to relate it to our subject. That is what is the mnemonic is all about. So one way is that, and one way is remembering it wherever possible, simplifying it. That is, don't try to go by methylene blue stains this, eosin stains this. So eosin stains only two things: granules of eosinophils and cytoplasm of RBCs in this context. And whatever else, if somebody asks you nucleus of neutrophil is stained by, so you go for methylene blue. Don't need to memorize so many things, these under methylene blue. So eosin, granules of eosinophils, the name matches eosin and eosinophils, granules of eosinophils and the cytoplasm of RBCs. Except this, whatever is asked, that is stained by methylene blue. Okay, so that is about staining. We will do it over there and count the cells. For counting the cells, and how will we count the cells and how to write the result, we will just show it over there. And this first try to identify the cells, especially those who are new, stain and try to identify the cells. And this will not come, you cannot become an expert in this in one day. So this topic, differential leukocyte count, we will do over, say, at least the next week. Okay. So what I strongly request and recommend is, if you have attended today's class and even you have counted all the cells, don't miss the next class, because still if you do not practice this for few days, you will not become very well oriented with it. Because the first issue will be in pricking the blood. Obviously, while pricking the finger and taking the blood, obviously while pricking your own finger for the first time, you will feel a bit anxious, and it will go away along with time. So repeat it at least few times, and this is one of the topics which takes, the total experiment will take, say, pricking the finger, taking the blood, staining it, then identifying the cells and counting them, writing the results. Total time, it will almost take 40 to 45 minutes. During the exam, 45 minutes is the allotted time officially, but usually we allow almost 1 hour for this. So this will be one single experiment during the exam for 10 marks. Okay. whatever marks are allotted in hematology for this one experiment, and the examiner will ask questions. Okay, so you to get it ready, one class is not sufficient. In one class, firstly you will the first in your life, the first blood smear you draw will not be perfect, not be a perfect smear. So a good smear, ideal smear is tongue shaped, right? It is tongue shaped. Why and how? Okay, we'll explain it as we go on and doing it. We'll explain it there. So please now leave your bags here. Don't carry your bags only. You may carry books and notebooks, and I will suggest, I strongly recommend that you carry practical textbooks. If all of you have not purchased till now, please purchase practical textbooks, physiology practical textbook by any author. Okay, those who have attended these classes before, I recommend the book by Dr. A.K. Jain because I find it suitable for the beginners. That is my opinion, but at least any one practical textbook by any author, at least for the hematology sessions, you carry it to the, in case of physiology, you carry it to the lab for any reference. Don't carry bags, only books or notebooks. ফিলটা বেশি পাবে 45 এ।
স্যার একটু আপনি মাইক্রোস্কোপ পার্টসটা একটু বুঝিয়ে দিবেন স্যার?
নতুন মাইক্রোস্কোপ
আইডেন্টিফাই স্যার একটা ওইটা
না, এটা ওভারস্টেনিং হয়েছে। হ্যাঁ, তো মাইক্রোস্কোপের পার্টস তো জানা উচিত। এই যে মেইনলি কি লাগবে, হচ্ছে এই এটাকে মেকানিক্যাল স্টেজ বলে। এই মেকানিক্যাল স্টেজটাকে, এর উপর স্লাইডটা থাকছে, এটাকে সরিয়ে দেখতে হবে। তার জন্য ফোকাস করার জন্য মেইনলি হাত দিতে হবে হচ্ছে দুটো জায়গায়, এই অ্যাডজাস্টমেন্ট স্ক্রুগুলোতে। এটা হ্যাঁ, এটা যতটা ওপর নিচের জন্য, যতটা এটা সাইডে আগে পেছনের জন্য, আর এটা হচ্ছে ওপর নিচের জন্য। তো রুলটা কি, এরকম করবে, মানে যখন তুমি এখানে চোখ রাখছো, আইপিসে যখন চোখ রাখছো, তখন কি স্টেজ থাকে ওপর থেকে নিচে?
সরি বলো আপনাকে লাইক স্যার, আমি একসাথে দুটো চোখ দিয়ে দেখলে ইন্টারাপশন হয়, মানে একটা চোখ দিয়ে দেখলে হবে?
ওটা কিছুদিন পরে ঠিক হয়ে যাবে। তো কারণ তোমরা হয়তো এর আগে হয়তো মানে দুটো আইপিস আছে এরকম মাইক্রোস্কোপ নিয়ে কাজ করো। আমরাও আগে করতাম যখন সিঙ্গেল আইপিস, তখন আমরা একটা চোখকে বন্ধ রেখে করতাম, কিন্তু দুটো চোখ দিয়ে মানে কিছুদিন লাগবে সেটা অ্যাডজাস্টেড হতে। ঠিক আছে, মানে অবস্ট্রাকশন কিছু হয় না, ওটা দিয়ে কিছুদিন লাগবে অবস্ট্রাক্টেড, মানে কিছুটা অভ্যস্ত হতে, কিছুদিন পর হয়ে যাবে।
ম্যাগনিফিকেশন গুলো স্যার কি?
ম্যাগনিফিকেশন তো যেরকম বললাম, এটা কত লেখা আছে, যত যেটাই লেখা আছে, 4টে আছে, তাই তো? তার মানে হ্যাঁ, যদি এটা দিয়ে যদি দেখি 10, 45 আর 100 আমরা ইউজ করি, জিরোটা মানে কিন্তু ম্যাগনিফিকেশন, এটা দিয়ে যেটা হবে সেটাই। হ্যাঁ, আর এটা দিয়ে হলে তখন কিন্তু 10x0 হবে না, বুঝেছো? এটা 10, তখন অ্যাকচুয়ালি ম্যাগনিফিকেশন হচ্ছে দিস মাল্টিপ্লাইড বাই দিস, কিন্তু ওখানে কিন্তু ম্যাথমেটিক্যাল মাল্টিপ্লিকেশনে 0 দিয়ে 10x0 হবে না, এটা দিয়ে যা ম্যাগনিফিকেশন হবে সেটা
আইপিস ইনটু অবজেক্টিভ।
তো আইপিস সেক্ষেত্রে মাল্টিপ্লিকেশন করে 10x0 তো 0 হয়, তার মানে এমন নয় যে নট, হ্যাঁ, তখন ওটা ম্যাথমেটিক্যালি মিল, কিন্তু বাকিগুলোর ক্ষেত্রে 10x, এটা হচ্ছে অবজেক্টিভ লেন্সেস যতটা ম্যাগনিফাই করবে, 10, 45 বা 100 হলে, এটা তো বোধহয় 100 ফোকাস করা আছে, মানে 100 এলে না 45,
সেটা আমরা ক্যালকুলেট
45 এর জন্য দেখা যাচ্ছে না, যে কারণে এই ফিল্ডে করা যাচ্ছে না, ওটা ওভারস্টেনিং হয়েছে। প্রথম হবে ওভারস্টেনিং, মানে স্টেন বেশি পড়ে গেছে, ওই গ্রিনিশ পুরো হয়ে আছে। 8 ফোঁটা, যেটি কোনো হার্ড অ্যান্ড ফাস্ট রুল তো নেই, যতটা স্মিয়ার ততটা কভার করতে হবে। প্রথম সাধারণত কি হচ্ছে, আকি দুটো স্লাইড দাও না, দুটো স্লাইড দাও, আরেকটা স্লাইড দাও। যেটা সমস্যা হচ্ছে, যখন আমরা প্রথমে করি, রুল ইজ যে এখানে আমরা এটা বসাবো, এখানে ব্লাড ড্রপটা থাকবে, এই হরাইজন্টাল সারফেসে রেখে এটা যখন বসাবো, এটা এখানে আছে, বসিয়ে ব্লাড ড্রপটার ওপরে জাস্ট আস্তে করে এখানে ব্লাড ড্রপটাকে স্প্রেড করতে দেবো। ডিরেক্ট বসাতে গেলে প্রথমেই ডিসরাপ্টেড হয়ে যাবে, হয়তো বাইরেও স্পিল করে যেতে পারে, তাই জন্য সামনে বসিয়ে পেছনে একটু দিকে এটাকে টানবো, ড্রপটাকে স্প্রেড করতে দেবো, তারপর ওয়ান স্ট্রোকে ইউনিফর্ম স্মিয়ার নিতে হবে। প্রথম যেটা হয় করতে গিয়ে বিগিনারদের এটা হতেই হয়ই যে ওয়ান স্ট্রোক আর হয় না, এতে কি হচ্ছে কেটে যাচ্ছে। এবার আমার যেটা লেখা আছে বইতে যে হচ্ছে কভার দ্য হোল লেন্থ অফ দ্য স্মিয়ার উইথ লিশম্যান স্টেট। এবার ডিসরাপ্টেড যেই হয়ে যাচ্ছে, তুমি কভার করছো, তুমি দেখছো এতটা স্মিয়ার হয়েছে, কি প্রথম হতে পারে একটু স্মল স্মিয়ার হলো, তুমি ওইটুকুই করছো, 4 ড্রপ লাগলো 5 ড্রপ লাগলো যা লাগলো, 4 ড্রপ 5 ড্রপ অনেকটাই, পুরোটা থাকলেও হয়তো অত 6 ড্রপ 8 ড্রপ লাগবে। তো এইবারে যখন করছো কিন্তু মাঝখানে তো গ্যাপ রয়ে গেছে, তার মানে স্টেনটা কিন্তু এর তুলনায় বেশি হয়েছে। না না, এতে একবার স্লাইড হরাইজন্টাল সারফেস, না হলে ব্লাড পড়ে যেতে পারে।
একটা জিনিস স্যার,
ওইরকম করে না,
স্লাইডটাই কভার করতে নাকি খালি স্মিয়ার টুকুন করলেই মানে?
না অবশ্যই অনলি স্মিয়ার টুকু, স্লাইড বেশি করতে গেলেই তো বেশি, মানে করে এক্সপেরিমেন্টালি দেখা হয়েছে যে জাস্ট স্মিয়ারটুকু কভার করলে সাফিসিয়েন্ট, তারপর আবার ওই প্রিকশন গুলো নিতে হয়, যে ফিক্সেশন, যে টাইমটা ঠিকমতো দিতে হয়, স্টেনিং টাইমটা দিয়ে লাস্টে যখন যে এক্সট্রা লিকুইডটা থেকে যাচ্ছে এরকম ভাবে ওয়াশ করতে হয়, তখন যেদিকে ব্লাড ড্রপটা ছিল আস্তে করে ট্যাপটাকে খুলে, মানে খুব জোরে নয় তাহলে স্মিয়ারটা ধুয়ে যাবে, আস্তে করে ব্লাড ড্রপে খুলে, হ্যাঁ আঙুলের উপর রেখেই আস্তে 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 এটা ধুয়ে যাবে। এটা ধুয়ে গেলে তারপরে আবার ড্রাই করে তারপর দেখা। এই যে কোনো একটা স্টেপে প্রবলেম হলে, যে একটা দুটো স্টেপে তখনই কিছু না কিছু, এইটা যেমন 45 এর জন্য কোনো প্রবলেম হচ্ছে না, 45 এ খুব ভালো দেখা যাবে, বরং ফিল্ড যত ম্যাগনিফিকেশন কম হবে তত ফিল্ডটা বেশি হবে। কিন্তু আবার 10 এ দেখলে অনেক, 45 এ দেখলে ভালোই দেখা যাবে, 3-4টে মানে 4-5টা হয়তো সেল থাকবে। এখানে দেখতে পাচ্ছি না, এইটা যেটা এসছে সেটা তো হচ্ছে ওই মানে ওভার স্টেনিং হয়ে গেছে বলে ওটা ওইরকম মনে হচ্ছে। প্রথম দিনই খুব, মানে প্রথম 2-1 দিনে খুব ভালো, 3-4টে ক্লাস হোক, একদম ডিফারেন্সিয়াল লিউকোসাইট কাউন্টটা পুরো বই থেকে পড়ে একদম ঠিক মতো করো, মানে স্টেপ বাই স্টেপ ভালো করে করার চেষ্টা করো। এবার স্টেনিংটা ভালো না হলে কিন্তু কাউন্ট করতে পারবে না।
স্যার এই
অয়েলটা নাও, অয়েলটা তোমরা আজকে তোমরা এর আগে করেছো, অয়েল দিয়ে করেছো, দেখো অয়েল দিয়ে দেখো, কিন্তু প্রথমে 45 দিয়ে দেখে নাও ভালো।
স্যার আমরা স্পেসিফিক্যালি হোয়াই ডু উই ইউজ দিস অয়েল?
এটা হচ্ছে রিফ্র্যাক্টিভ ইন্ডেক্সের এর জন্য, মানে ওই এয়ার আর তোমার তো এখান থেকে লাইটটা এয়ার আর গ্লাসের রিফ্র্যাক্টিভ ইন্ডেক্সটাকে এইটা ম্যাচ করায়, এই অয়েলটা।
গ্লাস আর এয়ার
এখানে কি অয়েল দেয়? সাধারণত সিডার রুট অয়েল থাকে, এখানে কি অয়েল দিচ্ছে? হ্যাঁ এইগুলো একটু ওভার স্টেনিং হচ্ছে, প্রথম এটা হবে, ওভার স্টেনিং হচ্ছে বলে এগুলো সব রাউন্ডেড। এটা এখানে এইটাই বুঝতে পারে, এই যে এইগুলো মোটামুটি মোটামুটি যেমন বোঝা যাবে এটা। তাও মানে এইটাতে দেখো যে যেগুলো বেশি পিঙ্ক এগুলো ডাব্লিউবিসি, কিন্তু ভালো ডিফারেন্সিয়েট করা যাচ্ছে না, কারণ স্টেনিংটা ভালো হয়নি। এই সবগুলো এই যে বাকি যে জায়গাগুলো আসছে এগুলো মানে রাউন্ডেড যে পিঙ্কিশ যে এটা থাকবে, এগুলো সব ডিস্টিংক্ট বোঝা যাবে, যদি যত ভালো স্টেন হবে তত বোঝা যাবে, সেটা একদিনে আসে না। সেই মানে এটা মোটামুটি স্টেনিং আর মানে বেশ ভালো হয়েছে। এটা তোমরা কততে দেখছো? মানে আমি বলবো যে প্রথম আজকে বা পরের দিন বা আগে যদি না দেখে থাকো, যদি 2 দিন দেখা হয়ে গিয়ে থাকে তারপর অয়েল দিও। পরের দিন এই স্মিয়ারটা যেরকম হয়েছে ওই ম্যাডামের কাছে জমা রেখে দাও, এই স্লাইডটা নিয়ে পরের দিন দেখবে। ঠিক আছে, আর কিছু না, কিছু এরপর এই তো আরবিসি, আর যেগুলো বেশিরভাগ আরো ভালো, মানে যেগুলো বেশিরভাগ সেল একই রকম, যেগুলো একদম বুক লাইক তো হবে না, মানে এইগুলো হচ্ছে আরবিসি, আর বাকি মাঝখানে যেগুলো এএসএল সেগুলো ডাব্লিউবিসি। কিছু আর্টিফ্যাক্টসও থাকবে, সেটা ওই ডাব্লিউবিসির ফিচার্স দিয়ে চিনতে হবে।